Yeah, I made like one new feature. But previous Sunday was like we have paper there. Tracer particle. There's like one or two. I think it's kind of making it. I mean, the range of particles. So I wrote down arbitrary here. I mean, that's here. So after you diagonalize.
Oh.
The laser as well, plug it in. Oh, that's what we were talking about before. Yeah. Would you like a laser as well?
Yeah, I think it's useful.
Okay.
Just in case.
The like slide back and forth.
Yeah. Is there a way to plug that in?
Okay. So this works.
Okay, that's
Okay, cool.
yeah,
We have to look to
awesome.
the left and right.
Yeah, that's good.
What was the new one you made? This is the center speed. Yeah. The one is more, sorry. Yeah. I don't want to close first. I just want to list it. And you open for the second one? Lecture? Okay, cool. Yeah, I'll be here for the first, so I can stay for the second as well. I'll come for the subset of. Cool, very good. Proper subset. Proper, yeah. Cool. About these lower.
Yeah, if you want water, there's water on the floor.
Oh, that's good actually. Yes, because you just had a lot of coffee.
Yeah, by the microphone if you didn't notice.
That's perfect. Oh, I see. Fancy.
Top
That's
shelf.
true.
Did you have at least good coffee? That's worse.
Oh, it was. I had Persian tea at the.
Oh, it's C4C.
Yeah, I really liked it. I wish I'd brought it with me. Yeah, I think.
I don't think there is a.
I don't know what's the. I don't know what's the base for it, but it was delicious.
I'm assuming it's some black tea, very.
Yeah, yeah, with some flavors of some kind.
I think it's the sugar cubes that make it break it. You know?
No, I think for me it's the dates.
I see.
When you have tea and dates, that's really does it.
Dates are-
Dates are really delicious.
I mean, they're surprisingly expensive in America.
Yeah.
They could say.
Yeah, yeah. And also, I mean, the food was amazing. You guys have a, if it's, I don't know if it's affordable or is it the right?
For the buffet.
Yeah,
It's
yeah,
about
yeah.
I think $11 now.
All you can eat.
Yeah, yeah.
That's crazy. Yeah.
No, we're just talking about this, just go there at lunch and stay for the dinner. It's even more affordable.
Yeah.
I guess, how was it in Stanford? The dining halls are good, or do you ever use
I've
it?
only been once or twice, like just with students if we go out, if we have a big group of undergraduates and yeah, it's healthy, but not like this where you can have cuisine from all around. It's just healthy,
Yeah, I think this
but
is
not,
this is
yeah.
rare.
This is like, oh, I can have Chinese food and Persian food at the same time. Mix and match. Yeah.
Well, I think how
I really like it.
homogeneous Boulder is. They have to make some sort of.
Guys, thank you for coming out on Sunday night. I'm very happy to have a new Prakash here, who has been sort of delocalized in many different places over the past month and made time to come out to see us today and tomorrow. Manu comes from Stanford in bioengineering, although a lot of his work, as you can see from his CV, now he's in biology and other oceanography related areas as well. So take it away, Manu. Floor is yours.
Okay, can you all hear me at the back? Yes? I mean, I have to say, you're not watching the Argentina-Colombia game. Or maybe you are. And if you are, please shout out the score, especially if Messi scores. I would, I think that's something that we can all celebrate. First of all, I have to apologize and thank many of the organizers. It's just, I haven't responded to emails for some time, and I do have one decent excuse. I've been at sea for the last 30 days, and so I am just kind of exhausted being at sea, but also I am happy to talk about what we were doing out there and what it meant, but it's just, it's a little bit backbreaking. So if you hear a little bit of exhaustion in my voice, I'm going to try to make up for it by good movies. But maybe before I start, I will just say this, of course, we spent 30 days at sea. I was looking for something, and we mostly study carbon sequestration in the ocean, and I might talk a little bit about it tomorrow. But there's sometimes strange things happen that you are just trying to figure out and put together in your head in science. In 30 days, I was doing a back of the envelope calculation. We processed around 100 million liters of water. So it's, it's mind-boggling to think about. It's like 50 or so swimming pools of water. And of course, the ocean is much bigger than that, so it's a tiny drop in the ocean. 30 days continuously every night for 7-8 hours of sampling. And but we were lucky enough to actually see a cell that has only been seen once in humanity's lifetime in 1906. And there doesn't exist a photograph, there just exists a painting. So of course, I was able to photograph an image and see the cell. So, I'm both tired and exhausted, but also just thrilled with excitement. The cell was the size of a big grape. That's a single cell. So, just giving you an example of why we go out on expeditions is just what we know and think about with biology and bounds of, many of you think about physical principles. How the hell would a cell that is literally a size of a grape, I could literally hold it in my hand. Many times we think that we know the principles. You can imagine, what would diffusion in this look like? Would it take a month for a message from the nucleus to reach? And I think we know so little sometimes of what is extremes of the living world that we really have to spend time and see. And I'm going to give you examples mostly from what are called non-model organisms to just share this perspective that often what we think are the bounds of biology and how we often think about how behavior works in living systems. I think it's quite limited. And another way that I think of this framework is, for the mathematicians, they have the number line, they have their abstract structures that they create. I think for us, at least in the biophysics community and the biology community, it's the tree of life. it really is a way that you can reach out and grasp and ask yourself, what would be possible? You can have a theoretical idea, completely abstract, and there is possibly out there an organism that is exploiting that mechanism or is exploiting that physical phenomena to its advantage. So I think there's a little bit of a tension in this sometimes, that you really do have to spend time exploring, but then you have a lifetime of analysis and thinking to do on those sets of systems. So maybe at the very end, if you ask me, I'll show you the photograph of that cell that has never been photographed. It's a beautiful cell. It's quite remarkable. Okay, so I think maybe what I wanted to talk about today is this, that I gave as a title, is just mechanical intelligence from a perspective of the living world first, and then we will sort of bridge that and think about, can we take these principles and develop much more fundamental frameworks in the abiotic sense to really think about where behavior comes from. And of course, a lot of people have been working in this space. I think I got started in this from the context of just physics of computation. For my PhD, I ended up showing that low Reynolds number is Turing complete, that you can truly build a computer even in Stokesian dynamics using nonlinearities and boundaries. And one of the things that kind of inspired me at that time is thinking about, what about other systems, and especially what we will call aneural systems, systems that don't have neurons? How would you think about encoding real-time behavior? And I'm going to share lots of examples. And literally, if you just think about the eukaryotic world, this is just a painting that I love. It's just, this is a figure from Corliss's paper. I mean, I just see incredible geometry in the eukaryotic world that I don't think we completely understand. We cannot really pinpoint from a form and function. And we've been thinking quite a lot about how geometry and topology in these systems impart behavior. I'll give you a few examples of these, and then we might move on from single cells to even animals that don't have any neurons, but have real-time behavior. And I think one way of thinking about this is also separation of time scales. Well, we're interested in behavior that are truly real-time, and hence the transcription translational machinery and the kind of information processing that happens in the nucleus, say, from a perspective of gene network regulation, that's too slow to give you real-time behavior. And what we've been discovering over and over again is that mechanics, and especially the cytoskeleton in many of these systems, could be thought of as an information processing system. And to the degree of complexity that you attribute to these systems is for debate. We can think about it. That is really a moving field at this point. But at least I am convinced that these sets of materials really have a kind of capacity that we have overlooked so far. Okay, so I think, and I'll give you a choice at the middle or somewhere once I'm done with this kind of a historic perspective, whether you want to start with the animal world and look at animals that don't have neurons, or do you want to start with the cellular world where we know they don't have neurons, but still have unexplained behaviors. But I think, just the protest out there, it's a remarkable number of organisms. And again, I just look at this and I often wonder, and many of these are planktonic cells. So that's why we go out to sea, because many of these systems you cannot grow in the lab. You really can only find them for less than a day or so, and then they die. And if you're really interested in behavior, you really have to be out there in an ecology. But if you were to ask me for any one of these, if I could explain, or anybody could explain these sets of geometries, it's quite hard. So I think they're missing sometimes this factor of just form and function has often been described as something that is quite coupled in biology. I just don't, what the hell is this? And over and over and over again, I can show you just remarkably beautiful architectures. And I think only when we go and look in this ecological framework, we actually find function. And so I think this is just also a call to action to many of you to bridge that gap. If you care about thinking about understanding biological function, just like you cannot understand any of it from the context of evolution, I would extend that to say that you cannot understand it without the context of ecology. That's really one of the biggest lessons that I've learned in the last 10 years, and that's made me leave the lab and go out in the wild. Okay, so I think one thing that I want to mention is, why has it taken more than 100+ years for us to even talk about behavior in non-neuronal systems? I mean, we see it all the time. Any of you who watched under a microscope, you've seen just remarkable things that cells do. And it was Jennings in 1906 in a very famous book called Behavior of Lower Organisms. And I mean, it was implying lower as if this is something less, which was the trend of that day. But you all know that most of these biological systems have had the same amount of time from an evolutionary perspective. So there is nothing lower or higher. They're just divergent and they're far away, but they've had the same amount of time. And he wrote this famous book. He was just, nobody believed him per se. He showed that some of these ciliates can learn. And actually, this experiment has been repeated now in 2 labs, both in Wallace Marshall's lab and Gunardhan's lab at Harvard Physics. And one of the things that started to happen at that time was an inkling of an idea that cellular behavior can have memory. There can be fairly complex things. But there was a sad kind of turn of events. And I think the story is quite circular. And that's why I really like talking about it. Jennings and some of the students, I mean, post-Jennings, really then started exploring behavior in single cells. And it kind of took off the wrong turn. And I'll explain to you that sometimes this can happen in science. Kofid and Sharp and several of the students went on to now then discover behavior in all kinds of cells. But in a strange way, because they saw the behavior that they could not explain, they started attributing and calling structures that they would see inside the cell akin to a nervous system. They were literally looking for a replica of a brain because we understood a little bit about that. Cajal had started thinking about neurons at that time. And they were actually watching cytoskeleton, but labeling it as the proto-nervous system. It became this theory of the neuromotorium. And they would sometimes see a certain density and they would label this as this part of the brain and that part of the brain. And it kind of took off to this crazy descriptive. And actually, ironically, they were discovering cytoskeleton for the first time. The structures are real, but they were attributing it function without understanding where that would come from, as if we are supposed to have a mirror of these higher, another hierarchy, which is multicellular. And then there was a Japanese group that came along, and then they started repeating, and nothing could be replicated. And that was devastating to a point that the entire field got shut down. And, I mean, in some sense, it's good it got shut down. But on the other hand, they gave us the cytoskeleton. And then all of these things got relabeled back again, where people started realizing these structures. But now, when the field of mechanics and mechanobiology came along, we started realizing that these structures can have function. And these structures can have functions in behavior. I mean, if you've seen some of the beautiful work from Wallace, and one of the threads around the cytoskeleton has really been that there are certain real-time functions that you can truly attribute to some of these cytoskeleton elements. So I think, I just want to give this as a historic perspective that sometimes fields have a false start. And then it's very difficult to work in that field. It doesn't mean that these questions are not good, but it just does come with a certain amount of baggage. You have to separate that out and you can start again. And I think over the last 5-10 years, some of that has begun. And I'm quite excited about what we have learned so far amongst many labs around the world. So I think I am going to make this projection in terms of thinking about this idea that you should boldly be able to stand and tell your neuroscience friends that you can actually do neuroscience without neurons, because neuroscience is really about thinking about animal behavior, but neuron as an element itself is not a necessity in that perspective. And of course, many of you have contributed to the idea of quantitative ethology and physics-based study of behavior. And that doesn't attribute what level of elements you implement that system in. And so I think it's quite a beautiful field. Now, the mechanics and intelligence and learning in mechanical structures in soft matter has become really another playground for you to play with. So this is, it's going to be exciting couple of years. Okay, so this was a side philosophical rant. I just want to say one more thing about at least how we think about learning and behavior in these sets of systems, which is slightly abstract, but it's worth talking about it. It's we've been calling this idea a perceptive machine, and I just want you to think about it in a completely abstract sense, not knowing the components. And this is related that in neuroscience, these sets of frameworks have been used. But in single cells and many broader systems in biology, this is playing out all the time, that there is some unknown internal state of a system. That's just any dynamical system could have that. And the world, which is the representation of whatever is outside this box, could be perceived and sensed by this system. And of course, that could change the state of the internal system. That's, you can have perturbations, you can kick it. And at that same time, you can also have action that can change the world. And what I mean by that is I could take that action and turn left, and suddenly the cues that I'm getting have changed. So there is this really interesting dynamical cycle associated with action, inputs and outputs, and essentially internal representation change. And this is really, now with this layer, you can go back and keep adding as many complexities as you want. And the reason I like drawing it this way is I cannot understand this system and its dynamics and what I might call computation in these sets of systems without the world dynamics. And often enough in computer science and many other fields, we want to make systems that are really not coupled to the environment. Many of my friends in quantum computing really want to decouple the environment because it messes up with your experiment. But often enough in a biological context, because the behavior is so tightly related to the attributes of function that have to be beneficial to the system, none of this could be understood without the world dynamics. And it's just an important, and hence you have to also understand how the environment changes by itself. And sometimes the interesting behavior that you care about is actually between these two systems, that there is a certain level of complexity here, but there is an inherent level of complexity in the world dynamics. For example, when we study life and behavior in the ocean is a stratified one-dimensional ecosystem. And what you do is reflected back and you find yourself in a new space and a new set of environment, and it becomes very important. And I think Matt and I, as one of the graduate students, talk about some of his work. One of the things we've also been doing in slightly abstract layer of this, and I just want to hint towards it because it's an idea that I'm excited about. This is not out as yet, but I just want to reflect it, that you can take these types of ideas, quite an abstraction, without even talking about a dynamical system. And one of the big things that we've been thinking about is this notion of, if we can have perceptive, if we can define without a physical attribute, different types of machines. And I think in neuroscience, they've done a remarkable job. There are different types of sets of attributes, but in this dynamical framework, we've been thinking about these ideas of what we call reflexive machines that really just respond to what happened at that moment, almost have no memory. Then we can make perceptive sets of machines. We can make attentive machines that have very long-term memory. And we have playful definitions of these types of, and this is all just made up. We have not as yet found sets of examples in which these types of, but we've been looking and searching hard for building an abstract language for these sets of ideas, which is very valuable because then I can attribute and not compare these systems to what a nervous system does. And this is one of the biggest challenges, which has been, is that often enough when you think about behavior, you think about it would be implemented in the same way. I mean, a lot of people who study cognition, even in octopus and some of these cephalopods, believe that it's not fair to actually compare these systems of what we have understood in the nervous system. So it's, this is quite abstract, but I've at least enjoyed, and I'll give you some examples of these when we get to the sets of examples. Okay, so that's abstract and philosophical. Let's actually go to the tree of life. I promised you that we are going to use the tree of life as a framework. So do you all want to be in the single cell realm? Do you want to go to the metazoan part of the tree? Where would you like to go? We can start with, we can go anywhere in this tree of life because we have examples from all across the tree, but I'm just giving you a choice. single cells and single cell behavior, or multicellular systems with complex behavior, but without neurons? How many of you are team single cells? And how many of you want to go to the multicellular world right away? Aha, you like your own kind. Okay, I'm just gonna, I think, I would say it builds up, and the multicellular systems have things that, I think it's layers of complexity, I would say. So I am going to try to cover both at least so that we have few of those. It's just, I don't know where to begin. Should we, I mean, our work started in this space from multicellular systems, and then we realized that we were missing in the cytoskeleton what was hierarchically somewhat similar. So, and I think, so in some sense, the progressive journey for us has been starting with multicellular and becoming smaller. But on the other hand, I think there is something, when you appreciate that a single cell can do this, then that appreciation allows you to truly think about what a collection of cells can do. Yes, Leo. Yes. Oh, I'm sorry. This is the number line for a biologist, effectively. And I mean, you know, I think one of the fun things about this is that there is a massive amount of tree right here that's just attributed and associated to what you would call metazoans, which would be multicellularity has evolved multiple times. Plants are multicellular and fungi are multicellular. These are animal cells, eukaryotic systems that join together and do not have a unicellular life. So just sort of all the way from here, you are here, and around this part of the tree. And actually, what I hate about the fact is the animal that I spent the most time studying is never drawn on the tree of life because it's so damn rare. No textbook actually draws it. And I've been trying to change that. It's been 10 years. I've written to all the biology, intro to biology, that please, could you include this phyla? Because it's as important a phyla as anybody else, and nobody cares. Because for the last 10 years, you know, it's close to sponges. There are 3,000 or 4,000 species of sponges known. And the animal that I'm about to show you, we have one species. And very recently, many of us have been able to now find 4 or 5 species. So, but still, it doesn't show up in intro to biology and most places. But you know, that doesn't mean it doesn't have something to teach you. And I think the other part of that is you can recognize some of the ferns and plants. You can recognize kind of the eukaryotic expansion. And I've removed the bacteria and others. I don't work on those things. And so I just, oh, this I would say, I mean, if you were to then be adding bacterial sets of systems, that's a massive scale again. If you just care about species as numbers, the bacterial world can overwhelm, not because of what we know, but because of what we don't know. So I think just to be very clear, we strongly believe that we have only seen maybe less than either 1 or 5% of life on this planet. So just in terms of what is not represented here is immense. And I think this 30-day excursion taught us that pretty flatly, that even just consistent sampling of one place over and over again explodes the unknown. Yeah, I think you should think of this as a very simplified representation. And I could find a tree that I could label with all the things that we worked on. Every one of the dot is, we have worked on that system directly. That's kind of, so I can only talk about things that I worked on, and hence that representation. But this is just, you should think of it as a cartoon representation of that number line. Yeah. Okay, multicellular. Okay, let's go somewhere else, and then I promise we will come back. I know it's somewhere here, so it's actually, in the yeah, this is interesting. Even in multicellularity, we could start with some. Yeah, I think in multicellularity, we might actually do 2D and 1D first. So let's start with 2D, and then I'll switch to 1D in an interesting way. So I think why do we think about these sets of systems is for a very simple reason, that neuron as an object didn't just appear out of nowhere. Neurons evolved, and there were animals that were out there that did not have neurons, because in a multicellular it's a very specialized cell. So how did they behave? How do we know? Ironically, when you look at the tree of life and the Cambrian explosion, that's the Burgess Shale where much of these fossils were found. All the phyla that we know today were actually represented in this Cambrian explosion. So that implies, and of course, many more phyla were represented that we do not have on the planet anymore. But what we know today about those sets of phyla, everything that's represented in the fossils seem to have nervous systems. And I think this was kind of a conundrum because soft-bodied animals, which were the starting point of this explosion, do not leave fossils behind. So when you think about our tree of life and our understanding of the evolution of the metazoan tree, it's significantly dependent on the fact that these things leave fossils. Now, there is a particular type of a fossil that we call trace fossils. Trace fossils are a trace, like a squiggly line that an animal left that you don't know what the animal is, but you can tell the complexity of behavior it showed. And these trace fossils are from unknown animals where the track was left behind and not the animal. So I think one thing when I started working in this space was this idea is, how do you go back in time? And the way you go back in time is asking these abstract sets of questions is, could we find an extant animal that exists today that does not have a nervous system or no semblance of neurons? And could we go back and ask how it works? We might be able to tell something about the past system. And that was sort of the starting point for us. And I think one of the things that I was excited about in this context is just how does computational capacity, no matter how you define it, scale with size of these systems? Because we believe that cells started clustering together. If some of you have seen work by Nicole King, the fact that one cell comes together, sometimes they stick. Sometimes when a cell is dividing, they don't separate out. So generating multicellularity is fairly easy. What is hard is whether it's useful or not. And so you have to think about that behavior. And of course, in neuroscience, you say, as the brain increases in size, somehow we are smarter. And actually, if you ask any day what we're doing to our planet, it's a clear evidence that large brain does not imply we're smarter. So that's very true. But unfortunately, kind of in this scenario, we still care about a scaling law for computation in these sets of systems. And I was lucky enough, almost 15 years ago when I started thinking about this question, that it actually turns out that there is an organism in the tree of life that has no neurons and no muscles. You know about sponges. Sponges don't have any neurons, but sponges just sit there. They don't do much, and I want to study behavior. So we stick with one organism in this tree, and you can see this is where you are. We're deuterostomes. Porifera is sponges. These are ctenophores. It's actually believed this is at the bottom of the tree of life. And this is the animal that I'll mostly talk about that people don't draw on the textbooks for a strange reason that I'll tell you about. So the irony of this is that we know that it has no neurons or muscles, but I'm just going to actually show you a first image to make you believe that this is worth studying, and I'll tell you about the history. So let's just spend a few minutes just watching this video. What you're looking at is a flat dish. There is a grain of rice for scale here. We like keeping living animals as scale so that this is roughly a grain of rice. This is a large dish, and I'm just going to play this video. The best way to appreciate this video is just hold on your eye to one single blob and just follow it. So here it goes. It's been sped up a little bit. It's a time-lapse video. And what you're watching is a clonal animal. They're all clones of each other. But you notice that I said it's an animal, and look at that string. It's still the same animal. It's the only animal that has no shape or form. So I know some of you like doodling. You can doodle anything you want, and in my database, I'll find an organism that is shaped like that. But of course, shape has a lot to do with how behavior might be controlled. And then I'm wondering if anybody has noticed something strange already in this film. Do you notice something strange? I mean, of course, it's walking around. We're going to talk a lot about what it's actually doing. The other reason we study this system is that we realized at that time that what you're watching is a pancake. I mean, some of you might have read that book, Flatland. This is truly a two-dimensional animal. It's 25 microns thin, and we can grow them in our lab to be 2 centimeters large. So that's an incredible aspect ratio. You can really think of this as a two-dimensional sheet. And I know, say that again? They are migrating just on a glass dish right now. And in this one, it happens to be that there is an algae mat and they feed this way. It's also the only animal that does not have a gut. So, many times it's said that in animal world, gastrulation is the most important event. They don't have a gut, so they eat by just sitting on their food. And the bottom sheet, so the architecture, which we'll talk quite a lot about, is take a beach ball, deflate it, let the two tops and the bottom of that beach ball fall into each other. That's going to be an ultra thin epithelial sheet. We call this epithelial because it's a continuous epithelium going around. And the bottom we're going to call ventral by tradition, the top is dorsal. And then we'll talk about periphery, which is joining. But it's a continuous sphere that's collapsed on itself. So anybody notice something strange? Do you notice something you can't explain? They flatten out, they pause, and that's actually a 20-minute clock, roughly, give or take. All of them do this. It's like there's a little shock that happens. Anything else? I mean, what is so bizarre about this, if you think, is that animals, we were told and taught, should have a shape. And this is the only example where shape itself is behavior, is the fact that when I said state or when I say memory, by transforming this organism, if any information is going to propagate, it will propagate in this very thin linear strand. And if some of you have heard about, we've been working on this idea of information bottlenecks in these systems. A thin strand can have an information bottleneck and propagation of information would change completely. So the state itself changes and they are utilizing this in this context to be able to do what they want to do. I think there was a hand right there. Yes. 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 And I love these sets of descriptions because, that's all we can do, because I cannot put my brain and literally feel what it is like to just crawl on a surface. My kids can, but I think this is what you have to understand, is that if you're studying a biological system and you're trying to come up with principles, you really have to be, what does it mean to live on a substrate to begin with? I mean, they can read an incredible amount of information from the substrate because they are absolutely in contact with it. Yes. That's correct. There is a stop and go motion, and we will actually show what is the mathematical principle of that stop and go motion. That is the one that we can actually explain. There's a lot of things we can't explain here, but that one we can explain. So, long story short, we started working on this. We built a lot of molecular tools to be able to literally see inside. Today, I'm just going to tell you a little piece of the flavor, but we've done a lot of work in the system. And just to really, why I got obsessed by this object 15 years ago, I rarely have a patience for one question or idea. This is the one that I've stuck with for the longest in my life, and that's embedded in this video. You're going to watch the animal, literally the left half says, "Thank you very much, I'm going to walk away from you." And that's behavior. It's as literally as if half of your cells said that we're going to go left, we're going to go right. Who made that decision? Right? I mean, somehow that decision had to be encoded, and you can see that that decision is being made collectively. And it's a very important decision because this is asexual fission. This is how they divide. So this is an active solid. I'm sure some of you have been working in this space, but this is an active solid that's kind of starting to make decisions on its own. This naturally then leads to a size distribution. And there is a finite size. I would literally, I'm not kidding you, because we are in Silicon Valley, some things like these happen. I got an email. We'd published these sets of papers about these animals, and I got an email from a person who's trying to start a burger company, wanting, "Oh, can we make this organism so large and grow them?" And I wrote to them, physics doesn't allow that, because I will show you a fundamental principle for why these systems become decoherent beyond a certain size, and hence they actually split. Yes. ruffled. Yes, and we will talk a little bit about that. We understand a little bit of that, and actually, but you have noticed something very important. This is an elastic sheet. So the sheet topologically over long periods of time in that movie, the connectivity must have changed, right? Otherwise, how can you split it? I mean, if all cell neighborhood remained the same, if it was purely elastic, you could not do this. So it's elastic in short time scales, and it's plastic in longer time scales. And the sheet actually fractures, which I'll show you, which is also important in this set of behaviors. So you have all of this elastoplastic behavior, and sort of we have shown that there is a brittleness in this system, that there are fractures that propagate that make these shape changes happen. And some of that curvature you're talking about is really coming from the elastic part of this. And the periphery, as you noticed, is a little bit, we believe, mechanically different from what's happening in the center. The cell sizes are much smaller. So anyway, let's zoom in a little bit to the system. And let me just skip. I mean, this is what I meant by when I said 25 microns, a thin sheet of an epithelium wraps around. I should just show you some movies, first of all. So what would happen if you were to see the cellular scale? And this is what I mean by how do we explain behavior in these systems? We built the sets of tools that we can watch every single cell in a living animal. And so just this is called in toto imaging. Like imagine if you're studying a system that you could see at least all the internal states. That was very important to us because who knows, maybe there is some strange thing that you're not observing. So you're going to watch a movie, and I love this movie. This is now the cellular resolution of that. You can watch, these are single epithelium sheets that are dynamically showing these contractile cycles. We show this is an actomyosin contraction. Actually, for people who think about actomyosin, we've demonstrated that this system holds the world record for fastest actomyosin contraction seen in the animal world. And there is no magic motor. It's actually in vitro reconstitution shows that actomyosin can do these really fast contractions. I think I have that slide. It's just the fact that the cell shape is designed to have very low load. But what's really fun is what I mean here. So now you're watching this sheet, and I've added one layer of complexity to it. And I'm going to add complexities in layers. This is the top epithelium, and any of you can see these remarkable waves that are propagating. These are contractile waves, and we showed a couple of years ago that excitability in the actomyosin cortex of a given cell can transmit. So if you have, say, a stretch sensor or a stretch receptor, when I contract my neighboring cell that is joined with a coherent junction next to it, you will feel that. And that by itself in an excitable system is capable of generating all kinds of wave patterns. So these waves are bouncing around. And I'm not going to talk about this. Much of this, the epithelial work is published, but I just want to tell you that this is the first layer of the active system that's happening on the top of this sheet. Yes?
These like edge modes, is this driven by something like odd elasticity?
I have to show you what's happening under, and then we will come back and talk about it. Yeah, there is a lot happening on the edge. I think one important aspect on this is that this is a short movie of a lot of behaviors. There are 3 or 4. There are spiral waves we have seen in this. We have seen these radial waves, and we also see these waves that propagate on the edge. But the challenge is that they are coupled with 2 other active systems. So this is an output of multiple active systems in this sheet. Okay, so let's just watch. Anyway, this was just to prove that this is an order of magnitude faster than any other system, but is reconstitutable. And this is just to give you another example of these sources and sinks of stress and strain. We can map them. We can really start thinking about what might be propagating this actually. So there's a lot of work that we have done in showing this, but I think the other thing that because I want to jump to connecting it to behavior, there is a second aspect of this, which is beyond a certain critical strain, it actually fractures. So this is now a living animal, a sheet. This is how it changes the topology, and you're seeing that giant hole that propagates. But also in the same amount of time, the system is able to repair the hole as well. So this is how it changes topology. And if you look at this sheet for a second, watch that sheet. There is the hole, and it's repaired. So did you all see this? Let me just play that one more time. It's moving around. there's the hole and it's repaired it. So this is like Terminator, but for real. And this matters because when you repair the sheet, the topology of the connectivity might change, and that's how you change shape. That you can break, like it's almost a brittle structure, and then there's a lot of simulations that we have done to show how something like that could change shape. Now we can do another class of experiments that we can really trap them in jails and watch these systems over a long period of time and study mixing. Because I showed you an example of this topology change, you can mix that. And this is one example of this. It's hard to see, but there is a jail, and we have tagged individual cells so that we can track them over long periods of time. I don't know which country flag it was, but imagine any flag that you paint on it, and you can track those sets of things to show exactly where those topological changes happen. And very importantly, it does not happen by the neighbor exchange mechanism that was discovered in Drosophila and then has been shown as the primary mechanism in morphogenesis. This is a fracture mechanism. So this is the layer 1. Now let's talk about the layer 2, and then we will kind of combine them together. And so this is the layer 2. And this was almost a tour de force of 7 years of work with one of my graduate student, Matt Bull. And the reason I'm happy you guys chose this because Matt went to undergrad to Colorado. So he always used to tell me about the summer school. So I'm actually happy to talk about his work here. What we discovered is that there is another mechanical system in this animal that demonstrates flocking. Very much kind of like the canonical flocking that many of you think about. And that is happening in the cilia. So I'm now imaging the bottom, and I'm just showing you certain class of patterns, and we're going to watch individual cilia, but these are flocks that are propagating information. These are spin waves. And we will consider these spin waves for the next 10, 20 minutes. But I just like admiring this because sometimes you are lucky and you can peer into the thinking of the system of what is actually leading to these sets of complex behaviors. So let's jump a little bit, and much of this is on archive. So, and we call this a mechanical brain because we truly believe that mechanics alone in this system is capable of generating complex behaviors, and I'll build this slowly. So first of all, we would like to understand how does the scaling work in a sheet if you add activity, how does that lead to behavior? Do we have simpler analogs of thinking about this? And I'm going to skip. I'm assuming you heard a lot about cilia from Christy already. Yes or no? Yes. I think the one word that I'll say for cilia is I really think cilia is like the hydrogen atom. I love it from a motility point of view because you can keep adding it and make a universe. In our life, we have studied all the way on the mechanics of single cilia, what they do in sheets, what they do in lines. How do you add defects in them? How do you add them as strings? We are writing a string theory paper for cilia of these guys who have beautiful strings, and just the behavior explodes. I mean, in your lungs, you have almost all organ systems. You're alive because you have cilia. And it's just such a remarkable, fungible system. And it's also the origin of behavior because LICA, the very first eukaryotic cell, actually had a cilia or a flagellum. And then we learned how to use them in 1,000 different ways. So it's actually very important in terms of thinking about behavior. And much of the single cell system's behavior is also driven by cilia. But long story short, what we were excited about is when we figured out a new technique to actually image animals in cross-section. So you're about to watch what's actually happening between the substrate and this sheet. So this is like a rubber sheet, very elastic, made out of cells that's on a surface. And now I'm going to play this. But rather than watching it from the top, you're watching it from the side. And this was really the first clear evidence that these cilia are not being used for generating flow, but they're actually walking on substrates. We can prove this with turf, that they are in contact, they are adhesive, as if these guys are wearing shoes. There is adhesion on the substrate. This is a high-speed movie that's been slowed down. You can watch the monocilia under, and you really see this new kind of a gate appear that had not been seen in single cilia before. And there are certain sets of other examples in single cells where there is a walking gait in cilia, so it was not that big a surprise for us. But eventually now you have to know that that's the second active system. So the top surface with actomycin, the bottom surface is active cilia. And now let's ask a question. How would you coordinate a million legs? Because there literally are million cilia in that tiny little animal, and you have to be able to behave in a coordinated manner. And this is where the story gets really interesting. So first of all, this is what the cilia look like. And we image the basal body, and I'll talk a little bit about the basal body is actually quite interesting. Basal body is the place where a cilia connects to a cell. And I'll show you that there is a new behavior that has evolved in the system to enable what you're about to see. But let's just first watch what does that look like in the context. That's the walking gait. Just like people have their classic power stroke and return stroke, there is a new emergent stroke. The second aspect that's important is if you watch the sheet carefully and we image very carefully, there are massive height fluctuations. So it is not a rigid sheet, it's elastic. And of course, it buckles. And when it buckles, height is a very sensitive nonlinearity that will become important in this system in a second. But let's just actually first take a look at the data, and then I'll come back to the walking cilia, because why did we care about this so much is because when we started imaging the system, so you can spend some time just looking at this. These are now single cilia, real time, and I don't know if some of you, if you noticed a vortex there. So I'll just play that one more time. If you notice right there, there's a little vortex that gets kicked out. So it's a quasi-particle of some kind. But this does not make sense from anything that we know about cilia, because we are told the way the cilia break symmetry is this 9+2, and it can only beat in that axis. That's really the power stroke. But here, what you're watching, you're clearly watching spin waves. So you notice that it was circulating, that got ejected, and that's actually a transition from rotation to translation, right? Because you are able to show if you can have this kind of and I like giving an analogy to flocking in birds, except in this system, there is no social force. There's no brain of the bird that's telling what to do. I'm going to show you this is all mechanics all the way down. That purely using mechanical systems, you can actually generate this type of coherent flocking. And now the complexity of that flock will determine the class of behavior. Yes? No, their topology of the system is not changing. That's a very good question. The cells remain in the same neighborhood in this time scale. In the longer time scale, as I mentioned, in minutes, they exchange neighbors. But I'll now play this a little bit 5x slowed down, and this is where that vortex is. And you can watch individual cilia walk, but what is really strange is many of you can see the circulation there, and that should imply that the power stroke in the cilia actually has a rotational degree of freedom. And then what we've been now able to show using cryo-EM, that actually the basal body has that structure that allows it to rotate over a certain time scale. So there is a torque term associated. There's torsional degree of freedom in a cilia, which is super exciting because in some sense, it is this new degree of freedom that now allows them to do something far more than just beat in the same axis. And this as a pattern is completely emergent. And now you might wonder, how are these cilia talking to each other? How would you know that this sets of an orientation needs to change one way or the other? And I'm curious if people have ideas. How do you think? This is all happening. This is 5 times slower, so this is happening incredibly fast. But if you have lots, and again, I'll give you a hint, which is an important hint, and then I'll come back. But this is what I want you to think about, caster wheels. What happens if you take a chair? Oh, go ahead, yes. Yeah, so I think the activity is in the cilia itself. That's the first energy injection in the system. But all of they are connected with the epithelium. And one of the important aspects is that there is elasticity. So the way to think about active solids in this case is the ciliary activity is injecting energy into the elastic structure. And then at a different time scale, and so this is kind of modes being excited, And then that elastic structure is injecting energy back into the ciliary field itself. So there is interplay between these two. And of course, one of the nice things about something like this is that you can write this down as has been written in active solids in some sense to be able to show that there is an explicit torque term in this equation. So this is an older formulation. I'll just show you this formulation for a second and then jump to show you all what does the actual cilia look like. We did this first, but then we realized we need to better understand what's happening with the cilia to truly explain the behavior. So one of the nice things about this is that this is very simple to write because there are no social forces. It's basically a torque term. And the torque term essentially comes from if you try this on your hand, if you slap your hand on a substrate that adheres to the substrate, but the rest of the cells are pulling you in whichever direction they're pulling you, you will see a torque term. And then you add a little bit of noise, and you can start creating a computational model of this. And there was a problem. And I'll see whether, actually, yeah, this slide that I had hidden, I should show you this, because sometimes a lot of ideas that you think about don't work, and it's the act when they don't work is when they teach you something new. So one of the interesting things that we discovered in the system that very quickly these types of systems will align and you get these beautiful ordered solids. But that's not what the biological system does. I mean, this just has this Goldstone mode, and now no matter what I do, how I perturb, it just remains locked in this system state. And what was missing for us was a very critical nonlinearity, which actually comes that we have to account for the buckling of this sheet itself. The fact that these compressive stresses lead to height fluctuations, and height of a cilia that walks is very important because if it is not in contact with a substrate, the force immediately falls to 0. And so that's the key nonlinearity. Now, if you were to think about this, if we write this down in the context of adhesion and elasticity, there is a cycle, very much like how people have written down forces associated with a cilia in a fluid. We can do this in this context, but with the context of nonlinearity of contact. And there is a phase space emerges, which is associated with if the adhesion is very strong, you're actually stalled. You cannot generate enough activity. You're just stuck. And then on the other hand, if you have absolutely no adhesion, you just slip on the surface. And then there is this nice space around walking. And one of the really exciting part about this is that these sets of sharp edges that you see is where these systems are excitable, that they can transition from one to the other with a very small fluctuation of height. And it just so happens that these types of systems actually have a parallel in how people have been thinking about, say, what a neuron does and why a neuron is excitable. Yes? Yeah, so the way the force is being generated here is that cilia has its continuous beat as an oscillator, but this set of cilia is adhesive. The way we know that is we actually do traction force microscopy on the substrate. We can see it applies forces on the substrates. We can also directly measure that adhesion force. But when in a ciliary beat, there is a phase in that beat which is in contact with the substrate. And then effectively, you can essentially transform this in a form where while it's stuck, it's the cell body that moves that makes it go from a phase where it's stuck. If I throw my hand here, now I am locked here, but remember this point is moving. You actually generate a buckling because the rest of the tissue is moving. So it's a type of a mode that only generates forces in the collective. And then once that has flipped over, you are back in that same phase and you make the next step. And one of the nice things about something like this is that it gives us an explicit term to write down as a function. We can write an analytical function here as a function of height. It's a nonlinear function for force of cilia. And there is an optimal term because if there is very little height, you can imagine you're stuck so tightly that you cannot do that clock. If you're too far away, you cannot generate the adhesion and you're out. And so I think this is really very important. And we needed these height fluctuations to be able to introduce the nonlinearities because any compressive stress actually leads to these fluctuations in height. And you can see this very clearly now if you go back and watch this movie again, you'll actually notice that the gaps are fluctuating. And then we do this all over the map, and you can see certain sets of, and that compression that you saw is actually a frustration mode where the entire wall collapses. And that happens because of a neuropeptide. And one of the nice things about that is that's the only way for the organism to stall. And when you look at that equation, it happens, just so happens, so I'm not making a grand claim here, but I just want to say that when we looked at these membrane height fluctuations, they actually match to some of the Fitsunagumo equations. And equivalent in neuroscience, there is a term of membrane potential and membrane voltages as a function of an ensemble. There is a classic mean field theory for that. And it just so happens these height fluctuations actually give you something similar. These are propagating waves. You can think of them as bucklings on a sheet. And it's the combination of these sheet fluctuations and the ciliary activity that gives rise to the behavior. And I'm just going to show one movie and then take the question. So now if you include those fluctuations, so now I'm not showing you the height fluctuations, and you can start seeing that the sets of behaviors that arise in these, they're able to generate these compressive waves that bounce back and forth. You're able to find a certain mode, but also come out of it pretty quickly. So the idea here in the context of behavior is that you want some amount of excitability, because otherwise you're just doing the same thing forever, that you're not taking inputs from the world. That's not a very good behavioral mode. But you also want some sense of coherence. You don't want to be so excited that you just keep shuttling around and then you have absolutely no coherence, because otherwise this massive set of a tissue can't do something repeatable. Yes? Yes.
So when you see the flagging behavior, is that basically kind of telling you that all of them needs to reorient or like get in phase in some sense?
Out of phase. Yeah, and then I'm not talking about this, but because some of you might have thought quite a lot about synchronization, we actually showed that this system has absolutely no synchronization with a lot of traction force microscopy and others. And one of the things that's important in this case is this idea that if you have coupled active systems, you can pump energy from the cilia in the strain of the sheet. So I'm now just plotting strain in this system, and you can watch all of these sets of stress patterns that are developing. And then these sets of stress patterns pump energy back into the ciliary mode. And it's this bounce between these two active systems is what's giving rise to the richness of the behavior. And then the other thing that's really fun in this computational context is for a certain amount of size where we can do the simulations. We can currently simulate around 30,000 cilia with this type of a mechanic. Just computationally, it gets harder and harder over time. But you actually see that these sets of coherent vortex structures, for example, so here I'm plotting as a size of the organism, this little region where these vortices are quite bounded. So coherence increases in this system until a certain point. And I think we do believe if we were able to computationally simulate some more, you would start seeing that the system starts breaking down. And that's really the idea of information bottlenecks, in just the fact that there are such large regions that are coherent with each other that they don't care about generating a massive coherent structure, which is what would lead to shape change. And of course, this system is very sensitive to shape, because if you ever get a pinch point, less number of cilia are communicating between each other. And if you have a thread connected them, they will get more and more independent. So effectively, shape couples in because of information bottlenecks. And maybe I'll just show you one picture, and then I want to switch to single cells. But what's really fun about systems like these is that they are far more complex than we give them credit for. Now what I've done is I've taken a very large animal. This is so large that it's not possible for me to track and watch single cilia. We are trying that. But what I'm doing is, because of optical flow of tissue is coupled to the flow of cilia, I can still track the vortex. And you're watching this vortex dynamics, and you notice that whenever it got kicked out, this is a pseudoparticle in the system, you transition from rotation to translation. But watch the first time. That vortex structure has wings, it splits into two, it joins together. So it's not as simple as just these two sets of states. There is a very dynamic states that these sets of points can exist in. And in larger animals, we've seen actually multiple counter-rotating vortices, for example, attracting each other, repelling each other. And also, there is an intuitive explanation we have for why these systems actually get kicked out when they come too close to the boundary. Okay, so that's all I want to say about the system. I could tell you a lot more. Yes, go ahead.
Are there enough, do you see enough defects in your system where like theories of active defect hydrodynamics might come into play? Or is it just like individual defect level where it's got hydrodynamics?
I think maximum number of defects we've been able to observe has been around 3 or 4. And that's just a function that the organism wants to do coherent things. But we have now very recently been able to introduce defects by a trick. And one of the things that allows us to then ask these sets of questions of whether some of the classical sets of systems would apply. I think one important thing to understand here is these are classes of systems where there is also noise, natural noise in the system, because the system is actually losing some amount of cilia too. So we see when an animal passes by, almost a bloodbath of cilia left behind. It's losing a certain percent. So there are certain kind of defects that are just geometrical defects. We've been able to now ablate some of those sets of zones. Another way of saying that is we can shave some of the legs. And then when we can shave the leg, I can generate those. So we've been able to make toroidal animals, for example, where the information only propagates in a ring, and then you're able to essentially generate behaviors that you would never see in a natural system. Yes? Yeah? Yeah. Yeah, I think there are two things we're doing in that space is we now have versions of the mean field version of this, because I think it was important to generate, in some sense, the height fluctuations. And now we know the wavelengths that are the natural wavelengths that should be relevant. Once we know that critical wavelength, then we have the capacity of starting to now write down very large sheets. And I think the other part of this is also associated with the fact that I'm not including another layer to the puzzle, which is now part of the next set of simulations, which we discovered recently that peripheral cilia in this system, which I didn't talk at all about, but I'll just show you a video, actually add another layer to the puzzle. Which is that the cilia that are down here is what I showed you the data for. But now we've been able to image the cilia on the periphery. And now you notice that they do a very strange type of a jamming behavior where they will form these hooks, and it's a way of hunkering down. They don't have the classic beats. And one of the things that we're now excited about is you take one class of a sheet, but you bound that sheet with another class of an active system. And most of these sets of cilia are very adhesive in nature. And this is also where many of the sensory cells are. So we've shown, for example, this system does thermotaxis. And so we can block that with TRP channels. We know that this is expressed in these peripheral tissue. So these peripheral cilia somehow play much more of a role of a sensory cilia. And then their beat patterns are not like these classical ciliary beats, but they often either jam, they will beat for a little bit, and then they will jam again. So there is another layer to the puzzle, which is associated with that. We should not be able to explain all the sets of behaviors because the boundary condition itself changes as a function of time. For example, in the system, we noticed very clearly that the ruffling that you had noticed is actually the seed where vortices get injected. So that sharp turn is essentially enough strain in the system that the vortex gets, that's the only way, essentially, that the system takes in a defect from the outside. Okay, I want to switch to single cells briefly. Yes, questions? Oh, go ahead. Yeah. Sorry, I didn't see you. Yes. Yes.
System, or is it like a separate
Yeah, it's a separate behavior, and we've been trying to induce it in our minimal model, and I think we need something else for it, which is that flattening happens massively like a wave, and it propagates from one place. And so right now, Charlotte and many people in the lab, we've been studying for the last couple of years the role of neuropeptides in this. So up till now, I told you a mechanical brain, but the story is even more richer than that, which is somehow this mechanical code is actually talking to a chemical code. And this is what I'm most excited about right now. And we're literally running those experiments as we speak. A few chain of amino acids, if dumped into this bath, will tell this animal to rotate. We understand how it rotates. We know it needs a vortex. But how can you have a chemical code talk and force a dynamical system to go into a certain mechanical state? And so there is another peptide that we can use, and that induces this massive slamming, and it actually comes and is induced by food. So the reason it slams, it's trying to essentially seal its periphery, because the food it's found will secrete enzymes, it will diffuse away, and you don't want all the food that you just digested to not benefit you in some sense. And that's also induced by a single peptide. And this is why the peripheral cilia have become very important, because we've been able to now identify which cells are the sensory cells in the system that respond to these peptides, and which implies that there is a chemical code that has evolved to be able to control a mechanical system. And I think the general thing I want to say here is in neuroscience, we don't appreciate how much of our brain is actually chemical codes because we can't see it. So there's all kinds of bursts of chemicals with diffusive dynamics, not the kind of long-range connectivity that's possible, but short-range diffusive dynamics that's at play. And in a similar system like this, the reason we're excited about it is allow us to really think about how chemistry and specific sets of cells could actually play as the right controllers of a large dynamical system. Where would those controllers be? And we believe those sets of controllers are on the periphery. And so now the kind of computational models we're building, and this is why we decided to implement behavior of peripheral cilia, because we notice when we add these sets of peptides, we see these induced on the periphery, and then they propagate. But it's a very interesting question of how does the chemical world and a mechanical world talk to each other, because we have a much better idea of the mechanics. But again, inside the cell, we know directly that those components are being modulated. So you have a direct link. Here, I just have ciliary dynamics, but cilia is a sensory organelle. I mean, primary cilia, all cilia are sensory organelles. They don't just sense force, they can also sense chemicals. And that, I mean, I think that's the space that has not been explored that much because we don't as yet know. I can tell you the probability of these sets of states in these simulations, but I can't tell you where you should turn the knob to make the system roll over and be stabilized in a certain mode. Okay, I'm going to switch to single cells. Yes? And we'll continue this discussion because I'm around for a long period of time tonight, so we can continue this. Maybe one thing I can mention is we got quite excited about this notion that you know, from a point of view of it really is what I showed you is just a very clever mechanical circuit. And one thing that we wanted to do is, you know, start to recreate these systems, because then I have true control. And as experimentalists, we get very excited about building these abiotic systems. So we decided to build this system that I'll tell you about. And the simplest way to explain it is imagine, so what we're now transitioning to is, you know, in this system, I can imagine many different ways of learning. But if I abstract it out and ask myself a question about purely mechanical systems, put those sets of networks learn, and the puzzle or a way of introducing this idea is the kind of learning that we're interested here is unsupervised learning. It's very important in a biological context. There is no hand of God that tells you that what you just computed is this far away. It's just, if it's not there, it's not there. And so it becomes very interesting to think about unsupervised systems. And one thing that we were interested in is in this environment and mechanics is this joke around, imagine your shoe changes as you walk on a given terrain. So when you go hiking, it becomes the hiking shoe. When you go running, it becomes a running shoe. It could read the environment, couple of steps, all injection of energies from your walking. How would you build a system like this? And while we have been thinking about making systems like this, artists have also been thinking about making systems like this. And that's just a conceptual, I don't know if I would ever wear that shoe. But the point around this is that adaptive systems can exist, but it was an interesting analogy to take a little bit further. Is it possible from an unsupervised learning perspective to truly build something that would be analogous to a neuron, but implemented purely in mechanics? And so this is what we wanted to do. And I think the best way to describe that is to just show you an example of what we are after first. So the simplest idea here is you can think about some of these neuronal systems in the context of what happens to inputs and outputs, essentially. So spike-dependent plasticity is a classical one. There is another kind of a rule in artificial neuronal dynamics called the Oja's rule. The object that we were interested in is something that would update its stiffness as a function of input that it sees to its periphery. Now, lots of springs out there will do something like that. That's not a surprise. But one of the interesting things about a neuron is its capacity to have directionality. Like the fact that there is the axonal body and there is the sets of places where it collects all that information, and then in the cell body, it'll make that decision. So there is a certain directionality to a neuron. And what we were interested in, can a directionality like that be implemented in a mechanical object? And what I mean by that is, I'll describe that very simply, is imagine a spring that changes its stiffness as a function of inputs it receives. Our bed does that, because if you sleep on one side, it kind of gets squeezed more and over time. So everything is adaptive, if you ask. But what's interesting here is we wanted a type of a spring that would only do that if, say, the information coming at the tail is larger than the head. And what I mean by this is that forces propagate reciprocally in this system. So if I tug on it, one side or it's not a diode, it's not a force or a mechanical diode, but its effective update rule has directionality. And I think, we arrived at this from this idea that to build truly a neuronal system, you really need something like this. Is this notion that you need a certain kind of directionality, that you should be able to differentiate what is a head and a tail in a mechanical spring. And I think this sat as a mathematical idea for the longest time, and we couldn't figure out how to build it. We actually convinced ourselves that it should not be possible to make it. But then Ian in the lab, graduate student, and Vishal, who's a postdoc, Ian is just absolutely gifted. He said, let me try. And we started making gizmos and started understanding them, while we had convinced ourselves before that it should not be possible. And we were thinking about these sets of springs, and we started adding certain components. I'm just going to show you this as a quick object, and I'll explain it to you. But I want to show you that mechanical systems can become very rich very quickly. I don't think it's about large number of components, and I'll show you why that's true when this directionality introduces a topological trick in these systems. So let's just watch what he built first. It's quite a puzzling object to watch. There is a little ring here, which you can see has a cutout. So it's thin here and stiff here. So if I could somehow rotate this ring, you will get the stiffness in this axis. It'll become soft, and it'll become stiff, and it'll become soft. That's also the state of the system. So the ring that you see here, if that ring could somehow rotate from the energy that's input in the system, you would get an adaptive spring. The adaptivity of the spring will be a periodic function, easy. But now, this is the puzzle. So we are shaking it from one end. There is some magical gizmo right there. There is a little pendulum that extracts some of the energy that drives the spring. you can watch this blue dot is turning, and you'll notice the head, when the object becomes stiff, you pass all the amplitudes. When it's very soft, the amplitude of the head should go down. So it's an adaptive system that is just rotating and going in the space of its stiffness. But if I take the same object and it's perfectly symmetric, and then I just flip it, and now I call the head the tail, and I shake it on the other end, you notice that it does not update. It's still passing the force because whatever the stable stiffness of this system is being propagated here, and it just happens it's in its soft state, so it's not overcoming friction. So this is one example, and I think it's the only known example of what I've been calling now a directional spring. So instead of drawing springs just as a wiggly line, in these systems, we also have to draw an arrow. And I think there's a long story. This is published work, so you can read how we achieve it. But the thing that I wanted to mention is once this can be done, something really strange happens. There's a very nice, clear explanation. By the way, this is the hidden magical object that makes that possible. There's a whole bunch of gears inside. There are actually 2 hidden dynamical systems. This happens to be a parametric oscillator. There are gating functions. So there is a mechanism that we were able to derive that actually shows how to implement this directionality. But when you have directionality, one really interesting thing that happens in mechanical circuits is this idea that topology of the circuit is important. How I connect them together starts to change. So now I have them connected in a triangle. I can connect them in a ring. You can see this one doesn't have a closed loop, and this will behave very differently if it had a closed loop. So we can really start doing circuit diagrams. And what's really interesting is even in a small set of circuits, I'll just show you. This is just fun to watch. You'll see all kinds of behaviors emerge that are dramatically different. Even if I change one single arrow in this system, white node is always the driven node. All the other nodes have some amount of friction. We use that friction to get the threshold function in neurons that you should not implement any motility until you reach that threshold. So that comes for free. And the reason I just brought this up, because I just want to say something provocative now, is this, if this was not provocative enough, is that it's possible that the cytoskeleton might have discovered this kind of a spring. This idea that you could have direction. We don't know an example of a directional spring in nature. It does really require internal degrees of freedom, but we've been starting to now think hard about, is it possible to have biological analogs of these types of springs? Because then very few number of components can essentially implement. And again, the whole reason we're excited about this is this system is capable of implementing unsupervised learning. So it can actually find patterns. We can train the system, very small number of springs. We can train them on a pattern and it can find them. It's not such a good pattern finder, so there is still a lot more work to be done. But at that same time, it is capable of learning. Yes. Yes.
What is it reading? So is it the diode?
So yeah, so what it's reading is the environment. So you make any arbitrary sets of springs, and you can either have input on one node or you can have inputs on many nodes. And all the other nodes have this friction component. And when you want to turn that into, in the context of learning, that, oh, if there is a pattern associated with the input nodes, the system will go into a certain state where it'll be an associated function. So one function, for example, could be is that one particular node in this network does not oscillate at all. Like it cloaks itself from all of the vibrations, for example. So I mean, everything here is sensing the directionality. Every spring is a directional spring. So, but what these types of networks allow us to do is that information propagates in these systems very differently than will be in reciprocal springs. And what's strange about this is it's not a diode. So it's not as if it's not allowing forces to propagate. Forces propagate reciprocally. The update rule is non-reciprocal. That's the idea. Yeah. And I think it's a strange idea because that's correct. Then it will decide whether if the head and the tail that we decided was higher, if the tail oscillation was higher, it will update itself. If the tail oscillation was lower, it will take the same force, propagate it like an elastic network, but not update itself. And that's really the heart of the Oja's rule. Oja's rule, and I mean, of course, if you think about the word back propagation has directionality integrated in that word. So there is a beautiful amount of directionality when you want to update. And like now we've been able to implement. And I mean, I had this long discussion with a friend of mine who makes quantum flux parametrons and kind of these squid-like circuits. And we could not agree what could be the electrical analog of this. So I'm actually quite excited for somebody to find, because we understand many of these systems in electrical analogs quite well, but I cannot pinpoint what the object should be. Yes. So in this case, the adaptivity is essentially the subtraction of the head to tail. That's it. So it's literally exactly the same as the Oja's rule is if the differential is 0, then you don't change your spring. If there is a differential, you change the spring. But then as you change the spring stiffness in the system, the information propagates in the entire network. So the optimization rule is actually not local because it propagates in the system, but effectively they will stop adapting if the differential, and if it happens that you find a certain place where the head amplitude is higher than the tail amplitude, then also they stop. So there is a directionality to that cost function, but you cannot answer that question for an isolated object that's embedded because its actual outputs are dependent on the entire system. Yeah. And it is not a classic energy minimization rule, for example. It's just an arbitrary rule. And I think kind of what's fun, I actually do believe that for many of these mechanical systems, it should be possible to generate all kinds of arbitrary rules and find new learning functions associated with it. Okay, I should say a word about single cells because I promised you, and it's only 5 minutes. So this is just going to be 1 or 2 pretty movies, but I want to have at least communicate that single cells can also do some remarkable things. So I'm just going to go really fast. And then I'm here, so I'm happy to continue and talk and chat. But anybody who would have to leave, by the way, who won the game? Does anybody know? Okay. Okay, all right. Okay, no, we will just continue. We can all watch the game later without telling each other the score. So anyway, I think one thing, there is one system that we study, it's called Lacrimaria Oler. We found it behind Facebook office in a swamp, and we've been studying it for the last 7, 8 years with lots of people in the lab. And I think we literally found it with a foldscope, which some of you might know is this origami microscope. I just want to show you this. It's a single cell. It just sits there, and this is data collected with a foldscope, and this actually has music. Although this music has been added. So although I feel like, the way it behaves. So that's what I mean by real-time behavior. What do I attribute this to? Like there are internal sets of things in this system, and I've paused the video now. I want you to focus on that particular thing, and that's a strike. And it's going to transform itself, engulf it, and continue. So if this was not scary enough, I'll show you the high-resolution version of this. Of course, we isolated the cell. We were able to clone them in the lab. We keep these massive cultures of thousands of them. And this is what actually happened in real time right there. That's the strike. Now that's DIC. And you can watch some, if some of you study cytoskeleton, you can watch a hint of what's happening in the cytoskeleton. So for the longest time, we were really interested in what encodes this behavior. And in a series of 3 or 4 papers, we have now shown mechanical components step at a time that lead to this type of search behavior. And so the first class of papers are the fact that shows that this search is quite blind. It's like when I was in grad school, I used to walk around and see if I can find food. And then, I'm not saying that you would do it, but it's kind of this notion of not caring about the information. And if you can forget where you are very quickly, you can actually generate a random search. And what we discovered a couple of years ago, using this very classical active elastohydrodynamic idea, that this kind of a system, so now this is an implementation of that system computationally. This is a follower force where the force is the function of the state of the filament. We can actually generate true chaos in a system like this. So what you're going to see here, for example, is a simple filament. This activity is turning on and off, and it becomes an oscillator. And this is a fun one. You can try staring at this and see what periodicity that you actually see. I am tuning elasticity and activity, and this is a 10-period oscillator, but it's a very strange one. If you notice the sets of counts, it is truly an oscillator. But when we put sets of numbers on it, for example, right there, that's the code. So it's 1, 2, 3, 4, 5, 6. It does that over and over again, but it's a pretty strange code, but it's generating a code purely. It's a very reliable code in this parameter regime that you could not have predicted traditionally, but that's what it is. It's a dynamical system because it has a lot of memory. The memory is in these buckling patterns, and these buckling patterns are very specific, so you cannot understand this without understanding these buckles. But majority of the parameter spaces, this is actually a random oscillator. It's actually a chaotic oscillator. And the way the organism finds food is essentially it exploits this idea of follower force to generate, just forget where you are, and you will hit upon an organism. And we showed that experimentally. We can match the kind of computational. So this is a one-to-one match between a biological program fed into a filament, and we can see these are the sets of simulations. This is the actual experiment. You can see that there is a zone that it is less well or less good at searching. These are sets of search clouds. So this was a hint that it's using chaos to search. Now, search behaviors are very common, but then came a second puzzle, and I think I'm just going to end here, but I just want to show you one picture associated with this. The second puzzle was that it also seemed to not follow the laws of mass conservation, which is not cool. No biological organism should do that. So you see a cell, this is 40 micron, and out comes this massive rod that just appears and disappears out of nowhere. This is real-time behavior, and no organism should be able to do this because you cannot make membranes, you cannot make cytoskeleton in real time. So there must be a storage somewhere. And the big question was, how could you actually have this happen in a single cell? And one of the things that we've been able to now show that it's a really, really clever trick. So first of all, you can torture these cells. And it's not torture because it itself extends. So a 40 micron cell, it's a single cell. 40 micron cell, I can stretch it to 1.5 millimeters. So like, what the hell is going on? And one of the beautiful things about this is actually using a trick from geometry. And it's a real, I think it's discovered this geometrical trick, and we now know from an evolutionary point of view how it got there. But one of the key ideas is, let's look inside. And what we discovered is that there's this beautiful cytoskeleton inside this system. So this is cortical microtubules, and this is a Z projection. So many people think it's a basket. It's not a basket. It's a helical coil on the surface of the membrane. This was the first thing that we discovered, but it has variable pitch helix, and the membrane is bound to it. And a lot has been studied on cortical cytoskeletons in some sense. But then we started, oh, by the way, that's the animal, the other cell that it's eating. And sometimes we find it on the inside. But we started looking much more closely with TEM. And because one of the challenges for us was microtubules cannot just appear and disappear. Membrane cannot appear and disappear. So the intuition was it stores it. But to store that much microtubules, you would need to make some kind of an architecture. And when we started doing these types of high-resolution confocals and 3D TEM reconstructions, we found that there are these 3 layers of storage. So somehow the coiling is not planar as it appears in that. And so now I'm going to show you TEM data. And so first of all, the membrane is stored in these massive pleats. So on the surface, it's pleated only till where the cell is the cell body. And when the neck comes out, the membranes become smooth. So somehow the membranes are pleated. Okay, lots of cells can ruffle their membranes. But now here comes the surprise, which is the microtubules themselves are ribbons, and they are on the top of the pleat. So this is one layer of this, what we call a microtubule ribbon. It's single microtubules in sheet-like form that are attached to the membrane. And what's super exciting about this, so that you can see the sheet, is I was on a vacation with my kids in Japan, and I wanted to see this artist. And I went, it was a paper artist, and we were, I'd been thinking about this for a while. There is an art of making a kind of a lantern in Japan, which is very old. And it's a type of an origami. And it occurred to us at that time that this system is actually using a curve crease origami to organize microtubules and membranes. And the idea is actually very, very simple. And the idea is this. When you take a helical-like structure and you make pleats on a cylinder, you can essentially fold and tuck in a lot of membrane. And what's happening here is that the microtubule is defining the crease of the fold, because microtubules are very stiff. So if you take any sheet of paper and paint it with a stiff pattern, wherever it's not stiff is most likely where it will fold. So between these pleats of a helix is where the fold is. But what's so strange about this origami, and we call this lacrigami, is a kind of an honor. This has never been discovered, this origami, is that it's a non-affine spring. A normal spring should stretch the same amount no matter where you are in a spring. And I'm going to play this video. We are pulling this spring made out of paper, and the blue is the pleat that was hidden behind it. And no matter what you do, it only folds and unfolds from a single point. It unfolds serially and it folds serially, which is, you know, when James Webb Telescope was sent out, we were on the edge of our seat. $1 billion, is it going to fold properly? That's just one fold. This organism is doing this 10,000 times a day. Every single time the fold is the same. And the robustness in this actually comes from a topological trick. There is a D cone, which is the point where the membrane is folded and unfolded, that is traveling along a helix. And as many of you know, in origami, if you have this traveling D cone, that's the only place the object can unfold. And in fact, in the organism, there are 15 of these ribbons. And for every pleat, because there is a valley and a mountain, there are actually 2 D cones. So there are 30 D cones that are traveling along, and they are coupled to each other. Because if one of them folds, there is higher curvature. You have these 30 D cones travel back and forth, and it's very much like if any of you go fishing, there is a spool. The edge of the spool is this transition from a nicely folded. How many of you have had your spool totally destroyed the first time you went fishing? Because the line cannot go back. It's because there is this singularity at the transition point of folded to unfolded. And no matter what, a good spool should only allow you to pack and unpack at that one point. And what's so strange about this system is the cell has discovered this geometrical trick. And then this is just one topology, which is this D-cone travel. There's actually another topological singularity in this system, which is a microtubule twist. And the way that you would see that is now I've made that same origami, but I made it with mylar so you can see it. And if I put, instead of microtubules, bamboo from Starbucks stirrer, and I've painted it on one side, you will notice that the pleat, when the pleat flips 180 degrees, there must be a twist, because what was normal vector facing down is now actually facing up. Does that make sense? Right? If I take a piece of paper and I folded it, you can notice that the normal vector to this is actually facing the cell. But if I open the pleat, this is facing up. And if this was a coil, at some point of time, the microtubule is facing down, the surface of that ribbon. And hence, there is also actually a twist singularity. So very much like in carbon nanotube or carbon nanotube ribbons, people have been studying what are called twistons. These are twistrons that are also traveling. And what they do, the D cone and the twistrons hold the energy. There is a little bit of an energy threshold that's needed to unravel it. And the only way it can unravel and pack back in is from this singularity. And so it's a really funny geometrical discovery that this cell made that allows it to pack and unpack an immense amount of material in a very short period of time. And one of the nice things that I like, and just as a clarification, you've all pulled straws. It is not a straw. A straw also has pleats, but they are not helically bound. So every one of them can open stochastically. But here, if you have this as a helix, there is only one way for it. There is no stochasticity in the system. And then once a while, very rarely, we can actually see the cell that generates a topological defect. And what has happened here is that the pleat opened at two places. The D cones were forced to open, and you will notice that now this bulge will remain forever. The system is not, because if you pull it hard at some certain set of a place, you can get another D cone to open. So now this has series of D cones here and series of D cones here, and they are folding and unfolding, and then they're folding and unfolding, and you can essentially very rarely get a defect like this. And then when we compare it to some of the other organisms that have morphologically the same shape, but they cannot do this transition, we actually find that, so this is another Diplytus. It has this behavior where it'll like an elephant trunk, but it cannot extend. And when you look at the microtubule, their microtubules don't have these coils. They just are straight structures. And so effectively, from an evolutionary point of view, all ciliates have cortical cytoskeleton, but just naturally, if this twist keeps winding and winding and winding, you will actually discover a mechanism like this. And I think this is how I feel like I want to end with this picture back again, that in every one of these pictures possibly is hidden mechanisms that are connecting geometry and topology to behavior that we as yet don't understand. So just like many people who study proteins deeply care about their structure, we should really care about the structure of a cell. And often enough, you will find a way connecting like every one of them and exactly where they're putting the sets of the cytoskeletal structure could give rise to a type of a behavior that would be attributed to that particular cell. So it's essentially a mechanical state space of geometry that they're exploring. And with each one of those geometries comes a completely new behavior. And I kind of feel that in some sense now, with the kind of tools that we have with expansion microscopy, we can finally see the cytoskeleton structures of these. So what was drawn here was what you could observe, but nobody could have observed without the TEM structures, the hidden microtubules, or you could not have observed the class of ribbons. So this is a really exciting time because finally microscopy is catching up with the complexity of many of these organisms. And I think we will definitely find lots of examples of these sets of geometries playing roles in behavior. Okay, I'm going to end there. And I'm around so I can take questions. We can all watch the game. But I should end on a note to just say, I don't do this work alone. There are incredible people, and especially all the members of my lab and collaborators that make much of this possible. Many of them have been out to sea with me, and they take on the sets of challenges of actually working outside the lab. And it's just an absolute joy and a blast to have adventurous people to work with.